see now what is the presentation.
Okay.
Yep.
So then I think that the most important persons are here, including the candidate, and we can start also this defense of a doctoral degree. In February 2020, Master of Technology Harsh Nitin Dongre submitted his thesis, Biomarkers and Preclinical Models for More Precise Diagnosis and Personalized Treatment of Oral and Vulvar Carcinomas, study on human samples and experimental models, to be evaluated for the degree Philosophia Doctor at the University of Bergen. The study has been performed at the Department of Clinical Medicine with Professor Daniela Elena Costea as main supervisor and Professor Line Bjørge and Professor Anne Kristine Johannessen as co-supervisors. The Faculty of Medicine appointed the following evaluation committee to evaluate the thesis. Professor Harry Hollema from the University of Medical Center of Groningen, the Netherlands. PhD Else Driehuis, University of Utrecht, the Netherlands. And Professor Havoye Miletic, University of Bergen, who serves as the chair of the committee. In accordance with the evaluation committee's report, the faculty has found the thesis worthy of being defended for the degree of PhD. And as required by the rules, Master of Technology Harsh Nitin Dongre have today given his lecture on the topic, the significance of tumor-stroma interactions for clinical behavior and therapeutic response in cancer. The evaluation committee has approved the lecture, and today the candidate will defend his thesis. Acting as opponents are, first opponent, Professor Harry Hollema, second opponent, PhD Else Driehuis. As this is a digital public defense, I wish to remind everyone present to please remember the following. Switch your camera off, as this may disturb the candidate and reduce the quality of the transmission. Mute your microphone during the entire lecture and defense. Do not test your equipment or functionality after the start of the lecture. It is possible to be granted permission to appear as opponents ex auditoria. Should anyone wish to do so, it must be made known via the chat function in good time before the second opponent starts. And we will also add a small 5 minutes break between the first and the second opponent. I will now ask the candidate to give a review of the objectives and finding of his scientific work. Please, Harsh.
Thank you. Acting Dean, members of Evaluation Committee, my supervisors, colleagues, friends and family on mute. Today I'll present my doctoral thesis work. The outline of the presentation will have introduction to cancer, rationale and aims of the study, results and discussion of all 4 papers with a brief summary and some future perspectives. So cancer is an abnormal and uncontrolled growth of cells that can arise anywhere in the body. Cancers can form a tumor mass and form their own blood supply. They can also invade and metastasize to different organs in the body, either through blood vessels or lymphatic vessels. This invasion and metastasis depends on both the genetic and epigenetic factors. In some of the cancers, mutation in TP53 is one of the first mutations that forms a patch of mutated cells. This patch or group of cells can acquire further genetic and epigenetic changes and form a tumor and metastasize to different organs. This flow of information from the genetics to epigenetics is governed by many RNAs that governs the transcription and translation. One such small RNAs, that is microRNAs, are 20 to 22 nucleotide long non-coding RNA that govern the translation of mRNA to protein. Based on their function, microRNAs can be both oncogenic, that inhibit translation of tumor suppressor genes, or they can be tumor suppressor microRNAs that augment oncogenic translation. Both of this results in tumor formation. So tumor is formed of more non-malignant cells than malignant cells. And some of the components of non-malignant or the tumor microenvironment are cancer-associated fibroblasts, CAFs, immune cells such as macrophages and lymphocytes, as well as endothelial cells that form the tubes. So as I said earlier, cancer is a group of 100 different types of cancer. In a research study combining 33 types of cancer, spanning almost 10,000 tumor samples, researchers have found a pan-squamous, pan-gynec squamous signature. So this squamous cell carcinoma were included. The type of squamous cell carcinoma included were head and neck, esophagus, lung, cervix, and bladder. And using this, they could distinguish squamous cell carcinomas from other carcinomas. However, this study did not include VSCC. So VSCC is vulvar squamous cell carcinoma that originates in the vulva, that is the external end of female reproductive system. And OSCC is the squamous cell carcinoma that arises in oral cavity and is included in head and neck, that is HNSCC. So if we compare common key concepts such as local recurrence, driver mutations, that is in TP53, FAT1, CDKN2A, PIK3CA, and HRAS, and look at the etiology-based factors such as HPV infection and inflammation, there are certain similarities between OSCC and VSCC. So given the notion of squamousness and other carcinomas, is this applicable to VSCC also? And if there is a squamousness in the epithelia, is it also applicable to the stromal component? So based on these rationales, the main aim of the study was to investigate the patterns of squamousness in oral and vulvar carcinoma with a special focus on tumor stroma. Further, specific aims were to investigate differentially expressed microRNAs and proteins in the stroma of both oral and vulvar carcinomas, and if there are differences, then to establish preclinical models to study this interaction. And a well-known etiology, as I discussed previously, is HPV, and it was to further use this established preclinical models to study the role of CAFs in HPV-dependent and independent carcinogenesis. So based on this, the outline of the study was divided into two cohorts, oral SCC and vulvar SCC. The first objective was to establish a microRNA detection in oral SCC. For this, we wanted to focus especially on tumor stroma, and that's why we decided to include protein IHC step. The protein IHC that was detected was pan-cytokeratin, which could be used to distinguish between the tumor part and the stroma part. So we did this, we introduced IHC and ISH methods by introducing first either ISH that preceded IHC or IHC that preceded ISH. In addition, we also did different combination of the chromogens for IHC and ISH and vice versa. All these methods, that is M1, M2, M3, and M4, were compared with M0, that is only ISH. So as you can see here in the representative pictures from M1 to M4, the staining of microRNA, that is in blue, and the staining of pan-cytokeratin, that is in brown, is similar to M0, not for brown, but for microRNAs. And this is counterstained with magenta. So further, we gave a guide to digital quantification using two different softwares. ImageJ is a freely available software, whereas Apereo ImageScope system is a paid license software. Using positive pixel area as marker, we saw that M3 method closely resembled the control method in case of microRNA 21, as well as in case of microRNA 155. This was also similar in Apereo image scope system, wherein it was similar in microRNA-21 as well as in microRNA-155. Since our scanning system was compatible with Apereo image scope and it was more or less more friendly than ImageJ, we used Apereo image scope system for further analysis. This analysis was done in OSSE cohort, and the cohort was suitable for biomarker discovery as factors such as age, tumor stage, metastasis to lymph nodes, recurrence within 30 months of primary treatment were unfavorable factors. When we did univariate analysis based on log rank test, we could see that higher microRNA 204 predicted a favorable prognosis in OSSE at tumor front. Also, even after adjusting for age and tumor stage, we could see that that microRNA 204 in tumor front was a significant factor. So to conclude paper 1, we have established a dual microRNA ISH and protein IHC method in FFPE archival tissue. Further, we gave a guide to digital quantification using freely available ImageJ and a Apereo image scope system. And using this, we have shown that higher expression of microRNA 204 in stromal cells is a favorable prognostic factor in OSSE cohort. Moving on, we wanted to see if similar or same microRNA expression is observed in VSSE stroma as well. However, when we did a pilot study consisting of 5 patients, we did not see microRNA 204 expressed in stroma. Next, we wanted to see if other candidates such as microRNA-21 and 155 are similar or not. And we did again in a 5-patient in a pilot study, and we could see that microRNA-21 was expressed, that is presented here in blue, was expressed in VSSE stroma. And microRNA-21 is known to be a poor prognostic factor in OSSE as well. Further, we used the M3 study from paper 1. And in paper 1, one of the drawbacks I could think of is that it stains all the stromal cells. But we don't know if it is particular for any specific stromal cells like fibroblast. So we used alpha SMA, that is a myofibroblast marker, and we combined it in M3 method. So now we have a single ISH and double immunohistochemistry. The immunohistochemistry or the marker, the chromogen for alpha SMA was magenta. So here you can see that again in pancytokeratin is the brown or the tumor cells. In blue is microRNA-21 and alpha SMA is in magenta. We quantified at tumor front and tumor center and observed that microRNA-21 is expressed more in tumor center and in tumor front as compared to surrounding normal vulvar mucosa. This was also true for alpha SMA and microRNA-21 in alpha SMA positive calves. So next we wanted to correlate with survival in VSSE cohort. Again, the VSSE cohort was suitable for biomarker discovery as factors such as age, FIGO stage, tumor size, body mass index, that is BMI, recurrence after 30 months, metastatic to lymph nodes, and tumor budding were significant factors. Apart from this, we also did immunohistochemistry on Ki67 and P53 that are common or classical factors in VSSE tumor progression. Using this, we could see that P53 expression or higher P53 expression was unfavorable immunohistochemical parameter. Apart from this, using the double IHC and single ISH protocol, we could see that microRNA-21 in alpha SMA in tumor front was a favorable factor. After adjusting to age and FIGO stage, we could still see the significance of microRNA-21 in alpha SMA in tumor front, as well as alpha SMA alone in tumor front as significant factors. Similar correlation were made with recurrence-free survival in VSSE. So here we can see that again, P53 was unfavorable parameter in the reference to recurrence-free survival, and tumor budding, even after adjusting for tumor size, was a significant unfavorable parameter in multivariate analysis. Same was true for alpha SMA in tumor front. This, we could conclude that we have established a method of triple staining, that is single microRNA ISH and double protein IHC. We also use this method to show that increased expression of microRNA 21 in alpha SMA positive calves predicts better survival in VSCC patients. And then tumor budding, that is more than 5 buds, is an independent predictor of recurrence. Next, we wanted to see why there is a difference. So we wanted to establish some preclinical models to study this tumor-stroma interaction. However, in VSCC, because of the rarity of disease, it is not very well established. So for this, we started collecting tumor biopsy from fresh tumor biopsies from patients, and we isolated till now we have isolated cancer cells, primary cancer cells from 4 out of 7, and out of this 4, only 1 was able to grow indefinitely. This cell line, VCC1, was isolated from VSCC that is associated with lesion sclerosis, which is a immunomodulatory condition. So we isolated this VCC cell line using explant method, wherein we cut the tissue into small parts and let it attach on culture plates. Further, we have isolated matched calves and to look at the tumor-stroma interaction. Lineage markers such as EpCAM and CD140B showed, or VCC1 were EpCAM positive and CD140B negative, confirming their lineage. Next, we wanted to look at the colony formation of VCC1 in comparison to already established UMSCV1A and UMSCV4 cell lines. So these are already established vulva cancer cell lines. And we could see that VCC formed significantly more number of colonies and more number of spheres as compared to other established VSCC cell lines. In addition to this, VCC1 was isolated from HPV negative patient or tumors associated with HPV negative phenotype. And we could see that even in DNA, as well as in P16, which is a surrogate marker for HPV, that it was HPV negative. Gene expression analysis resulted in interesting observations were made in gene expression analysis, where it presented more EMT-like phenotype in terms of cadherin-1, E-cadherin-1 downregulation, as you can see in red, whereas upregulation of twist1 and vimentin, that is in green, as compared to other UMSCV cell lines. Next, we wanted to see the role of fibroblast in invasion and tumorigenic potential. For this, we grew VCC1 on collagen-containing VCAF and without VCAF at a liquid air interface to mimic the differentiation pattern. Using this, we saw that VCC1 did not invade the matrix in absence of fibroblast. However, they invaded matrix even in presence of normal fibroblast or cancer-associated fibroblast. In vivo mice models, when we injected VCC alone, it did not form continuously growing tumors, whereas we co-injected with the VCAF, they formed continuously growing tumors. And the histology of co-injected tumors was much more similar to the patient tumor from which the cell line was derived. This was also similar in characteristic keratin pearl formation, invasive single cells, muscular invasion, and perineural invasion in between the patient tumor and the xenograft tumor. So to conclude from this part, VCC1 is a novel and unique cell line established from VSCC associated with VLS. It exhibits characteristic epithelial morphology with increased stemness and EMT-like phenotype in vitro. In vivo, the enhanced in vivo tumorigenicity is associated with CAF, and it suggests a critical role of CAFs in VLS-related oncogenesis. Moving on, we wanted to use these preclinical models to study role of CAFs in HPV-associated VSCC. The major subtypes of HPV in VSCC are HPV 16 and 18, apart from 31 and 33. So logically, we overexpressed E6, E7 oncoproteins that represents HPV-associated phenotype. So when we did that in two different cell lines, BCC1 and UMSCV4, we saw that in UMSCV4, it increased proliferation, colony formation, and migration, whereas in VCC1, it did not have similar phenotypic effect. Upon NGS analysis, we could see that VCC1 had a gain of function T53 mutant, whereas UMSCV4 had a functional mutant. This could suggest that the E6 oncoprotein could bind to the functional T53 mutant and carry out its oncogenic effect. However, these are just preliminary results, and further studies are required to confirm this hypothesis. Next, we wanted to see how calves respond to cancer cell co-culture. For this, we used a non-contact transwell-based co-culture system in which we grew fibroblast in the lower insert, whereas we grew cancer cells on the inserts. Using this, we could see that in co-cultures with SCV4 wild type and E6E7, VCAFs produced more HGF. When we wanted to look at the cancer cells that were grown in co-cultures with fibroblast, we saw interesting results. Apart from different markers such as c-Met, TGFR, TGF-beta receptor 2, we saw increased PDGFR beta and VGFC. So now these were interesting results, and I will follow them differently. First, I'll start with the PDGFR beta and then come back to VGFC increased response. So this gene expression analysis was confirmed at protein level also via immunoblotting. And interestingly, we could observe that in wild type co-cultures, there was increased phosphorylation, whereas in E6, E7 co-cultures, there was decreased phosphorylation. So now phosphorylation of YAP is related with cytoplasmic retention and degradation, which was confirmed by immunofluorescence, in which we could see that in wild type co-cultures, YAP was not in the nucleus, it was more in cytoplasm. Whereas in E6, E7 co-cultures, it was more translocated to the nucleus. So upon analysis, it could be seen that in UMSCV4 E6, E7 co-cultures, it was in 80% of the cells, whereas in wild type co-cultures, it was less than 10%. So what is happening here is in both wild type and E6, E7 co-cultures, PDGFR beta is upregulated, but only in E6, E7 cells, the cytoplasmic YAP is translocated to nucleus. Again, these are preliminary results, and we need to verify them further. So as a result of YAP nuclear translocation, we could see increased migration in scratch wound assay. But this increased migration was observed in direct co-cultures, wherein we grew fibroblast and both wild type and E6, E7 cancer cells together. Whereas in indirect co-cultures, where we took conditioned medium from the fibroblast cultures, it did not have any significant differences. Similar results were obtained in transwell migration assay, where E6, E7 cells migrated more towards the fibroblast, but not wild type cells. So this result suggests that bidirectional crosstalk is important between CAFs and E6, E7 cells. Next, we wanted to see how it behaves in a 3D organotypics. So while wild type cells did not invade in matrix that did not have fibroblast, or even in normal fibroblast or CAFs, E6, E7 cell invaded only in presence of CAFs. So this is interesting, as gene expression analysis show that there is an increase in MMP1 and MMP9 that is upregulated in this condition only. Again, these are preliminary results, and we need to do some MMP degradation assay. Moving on, when we injected both UMSCV4 wild type with CAFs, the CAFs enhanced in vivo tumorigenicity in both wild type and E6, E7. However, this effect was much more pronounced in E6, E7. Now, coming back to the VGF story, so increased VGFC is a factor that is responsible for pro-lymphangiogenetic response. So again, this was confirmed by ELISA, that is enzyme-linked immunosorbent assay, in which we could see active form of VGFC. So in this, the increase in the VGFC expression was not increased in wild type cells. However, when grown in co-cultures, the concentration increased, but still the increase was not significant. In terms of E6, E7 cells, they already have a very high baseline VGFC secretion, which was increased at, which is a time dependent and increased to almost 500 picogram per ml. In case of E6, E7 co-cultures, the expression did not increase at 6 hours, but as time went on, like at 12 hours, this increase was significant to the wild type co-cultures as well as to E6, E7 alone. This was further supported by immunoblotting data, where in the receptor for VGFC, VGFR3, is upregulated only in E6, E7 co-cultures. So VGF3 is a very important molecule that plays an important role in sprouting of lymphangiogenesis vessel. So all this data supports the idea that E6, E7 cells have a pro-lymphangiogenic response, but again, further validation is required. So in order to test in vitro lymph tube formation, in order to test the functional significance, we tested it in in vitro lymph tube formation on matrigels. So when wild type cells were grown in absence of fibroblasts, it did not form tubes. Whereas E6, E7 cells, because of their increased VGFC secretion, they could form stable tubes. When calves were added, the stability of wild type cancer cells to form tubes was increased, but this was not in a time dependent manner. However, in E6, E7 cells, increased calf co-culture increased stability even at 12 hours. I think it would be much clearer if we look at the time-lapse video. Yeah. So here you can see in wild type alone, the tubes are not stable and they degrade faster. Whereas in V calves, the tubes are stable and they last, or they are functional as compared to wild type. In E67, although they are more stable than this, but they have a leaky vasculature, which could be associated with cancer. However, when E67 cells are grown with calves, they are more stable and they form this elongated cancer cells. We still don't know if they are functional, but they appear to be tube-like structures. And Z stacking showed that they have a lumen, but still we are not sure if they are functional, and further assays are required to test this functionality. So to conclude, in co-cultures, we have seen that in E6, E7 cells, there is an increase in V calves. In both wild type and E6, E7 cells, there is an increase in HGF, but this increase in HGF leads to sharp nuclear translocation, either via PDGFR beta activation or c-Met activation. This leads to proliferation and migration, and it could better YAP nuclear translocation could stabilize HIF1 alpha and increase VEGF-C, causing lymphangiogenesis. In 3D organotypics, we have seen that in co-cultures, they secrete MMPs that degrade the matrix and increase invasion. However, these are still initial hypotheses, and we need further results, further experiments to confirm this. So now to summarize the whole thesis, we have established a multiplexing protocol to study squamous by combining microRNA ISH and IHC. Using this, we report that higher expression of microRNA 204 in stromal cells at tumor front is a favorable prognostic factor in OSCC. And using a modified protocol, we found that increased expression of microRNA 21 in alpha SMA positive calves predicts better survival in a subset of VSCC patients. So next, to test the hypothesis of squamousness in comparable cohorts of OSCC and VSCC, we see a distribution of expression of epithelial markers such as P53 and K67. As similar, but in case of alpha SMA, that is the stromal marker, the expression is very different. This also reflected when we did Cox regression analysis, and we saw that the pattern of K67 and P53 was similar in OSCC and VSCC in overall survival as well as in recurrence-free survival. However, alpha SMA, that is significant in both OSCC and VSCC, showed different results. So in OSCC, it was unfavorable, whereas in VSCC, it was more favorable. Even though not significant in recurrence-free survival, but we could see that it is more towards unfavorable prognosis, whereas in alpha SMA, in VSCC, alpha SMA is a favorable prognostic factor. Apart from this, we have also developed preclinical models to further biomarker discovery. And we have established a novel cell line VCC1 and isolated matched calves to study the tumor stroma interactions. And we have used this to show that E6, E7 cells have a pro-lymphangiogenetic response to calves. In future, continuing the biomarker discovery, we would like to use imaging mass cytometry to compare the different markers in OSCC and VSCC to establish the concept of squamous and to study role of calves in HPV-associated VSCC in respect to YAP signaling and how it governs tumorigenicity. And in addition to this, we would like to gain some mechanistic insights into this E6, E7 dependent lymphangiogenesis by vulvar cancer cells. I would like to thank my supervisors, laboratory facilities that I have used, funding agencies, and my lab mates that have maintained a very conducive microenvironment. Thank you for your attention.
Okay, thank you so much. I thank the candidate for this review of his work, and I will now ask the first opponent to commence. Please, Harry Holleman.
Very nice presentation. Being a pathologist, when I teach, the students always ask, will there be after, say 5 to 10 years, or can we rely on all the things you demonstrated? What do you think?
Sorry, Harry, I missed some of your.
Sorry.
Yeah, I could not hear you.
When I teach the medical students,
Uh-huh.
and they always come up at the end of the lecture with the question, the pathologists are outdated, and we should rely on the work you presented. What do you think? Just next generation sequencing, just measuring tumor cells in the blood, to name a few.
Yeah, so actually where I work, I'm surrounded by pathologists, so I would like to answer it in a more diplomatic way.
Yeah.
So I would say that no, pathologists are important because they are the ones that define what processes to be used. Like if you give a pathologist any slide without saying what it is, then he would not know. So you have to rely on pathologists to suggest the further ways like NGS or calculating CTCs in blood. So I think pathologists play a very important role.
Okay, thank you, because we're still training pathologists, and they come up with the same question: what will I be doing within the next 30, 40 years? Eh? Still, you write on page 4 of your abstract, conventionally, cancer treatment is based on the site of origin and not on morphological or molecular evidence. I do not agree on the morphological point, because that's my... Would you comment on that? Because morphology is very, very important, I think. You mentioned there are about 100 types of cancer. I always teach my students there are several thousands forms of cancer. There's not just one lung cancer or one ovarian cancer or one brain cancer.
Yeah, so by that statement, what I meant was like the cancer treatment that is based mostly for carcinomas is based on the site. That is, if it's in oral, it's mostly surgery followed by radiotherapy, whereas if it's in vulva, it's again the same. And right now, there are very few studies. I think with PD-1 coming up, there are few cancers that they just test for PD-1 and then the cancer treatment is decided. But apart from this, I don't know any molecular markers or yeah, morphology is again important because that decides if carcinoma is squamous cell carcinoma or adenocarcinoma. But apart from PD-1, I'm not sure if there are other molecular evidences. Like at least to my knowledge, is there anything else that that you know or you suggest?
No, I agree with you. You mentioned the anti-PD-1 study. It's in HPCC patients that will, and those patients generate a lot of new neo epitopes. So that's why, in general, HPCC-related cancer is more likely to respond to immunotherapy. I think that's the only one I know of. But also, most of this type of work you presented is very nice and very interesting in generating new therapies, but it's also very difficult to reproduce them. You've of course read the papers that says only 15 to 20% of all these studies that are being published are reproducible, and I think but that's also due to the fact, for instance, ovarian cancer that can be divided in several subtypes, and they never have enough, and they always talk about just one type of ovarian cancer, and then your results are very dependent on the several types, and they have different pathways to carcinogenesis. So you need international cooperation to generate enough cases to get reproducible results. What do you think about that?
Yep, like all the new technologies like NGS or CTCs, they kind of generate new data, but if you look at the epidemiology, it's not sufficient to predict in a lot many patients. So I still suggest, or I still think that you can predict for a specific subtype or specific mutation, but to predict for all types of cancer, there's, just like you said that for ovarian cancer, there are different subtypes. So it would be difficult. But looking at just the histology, oral squamous cell carcinomas and vulvar squamous cell carcinomas are predominantly SCCs. So maybe in that it would be better. But yeah, we have seen that in OSCC, it's a very heterogeneous group. So it depends on what pathways are upregulated, and it needs much more evidence actually.
Yeah, I agree. Just for instance, vulvar carcinoma, you have at least two types, maybe more, HPV dependent and HPV independent, and the majority are HPV independent. And maybe there are several subgroups, P53 positive or mutated and not. So, but it's difficult to get enough cases. You're right. And it's also something that intrigues me. The number of patients with vulvar cancer is going up, and you say that's maybe probably due to an increase of HPV infections, but I don't see that in our cohorts. We have several hundred of cases of vulvar cancer, and we have about, a little bit more than 20% HPV, and it's constant over the years. So any other explanations why?
Yeah, actually, I would say it depends on the cohort again. So mostly HPV dependent BSCC are in early age women, and most of the cohorts that I've seen or that I have referred to are old age, and that's why they have this keratinizing type or HPV independent carcinogenesis more. And apart from that, there are HPV vaccines available, and it is kind of compulsory in the Western world to have both the men also now, and both the boys and girls vaccinated. So that could also lead to decrease in numbers. But again, I think so it has been in practice for 5 years. So if you take into that consideration, it will take at least 20 years for the HPV number to go down. But yeah, most of the codes that I've seen are in 60, like that the median age is 65 or above. So that's why most of them are HPV independent.
Yeah, I agree, but it's also intriguing why in vulvar carcinomas, also the women with HPV-related cancer, it has a very, very broad range. We have seen patients from around 20, but most of the HPV positive cases are also in their 50s. So why, where do I get the HPV from? Because for cervical cancer, you see that 2/3 of the patients are around 30. That's in line with acquiring HPV during intercourse, and then they develop cancer. And why do you develop cancer, oral cancer, or vulvar cancer at a late age, also HPV related? I do not understand. Do you have any idea?
It could be that there is HPV association, but it is not enough to drive oncogenesis at young age. And maybe as I explained in my trial lecture, it could be that the microenvironment is more tumor promoting, and then the HPV could drive oncogenesis on its own. And there are studies that have shown that only HPV could not initiate oncogenesis, but it requires mutations in different pathways. And I think with old age, not only the pathways, but like there is a synergistic effect between the HPV association and different upregulation of different pathways that could explain why it is also in old women.
Might be, but and then the squamousness, it's a big part in your thesis.
Yeah.
What does it help us? I see more differences than similarities. You can come up with a nice concept, but how does it help us? Why is it important?
Yeah, so right now I've focused, or most of the papers have focused on the epithelial component of squamousness, and it would be important, right? Let's say for like PD1 expression, if it is present, then the patient is given anti-PD1, or even if it's not, in some cases it has been shown to increase survival. So what I think is, if all the squamous cells behave similarly, or there are some pathways that could be targeted, then irrespective of the presence of, let's say it's in oral, or if it's a lung squamous carcinoma or vulvar squamous carcinomas, it could be given a same treatment. So irrespective of the site of origin, defining squamousness would help in giving a broad treatment.
Yeah, but I may be stupid, but I think it's a very difficult concept, squamousness. Can you explain to me what squamous actually is, apart from the idea that some squamous tumors share something? I find squamous a very difficult concept.
Yeah. So it could be that some of the squamous cell carcinomas or squamous cells, they require similar pathways that could upregulate themselves, or that could be carcinogenic. I think to answer your query, it would be, I think the tumor stroma plays a major role. And even if we target squamous cells independent of origin of site, we have to keep in mind the stroma, especially in females, where it is almost bathed in sex hormones or just hormones.
Okay, maybe let's go to paper 1.
Yeah.
You write in paper 1 that cancer is a dynamic disease.
Mm-hmm.
That's true. It does not stop growing, and it changes all the time because there's tumor progression. And this heterogeneity can be due to genetic, agree, epigenetic, agree, transcriptomic, okay, and phenotypic changes. I think the phenotypic changes are a result. It should not be in that line.
Yeah, I agree.
Agree?
Yeah.
Because, that's
because the phenotypic changes are due to all the epigenetic changes.
Yeah. Okay. Then, you say common molecular alterations. Okay, we came already to that about squamiousness. That's. I see a total different carcinogenesis in vulvar carcinoma and oral carcinoma. And some of them share HPV, but okay. Do you think in oral carcinoma, I'm not a specialist in oral carcinoma, but you write that dependent on site of origin, there's a difference in behavior. Did you see any difference in the stroma of the sites, or can you explain it? Why tumors on the tongue or on the deeper oropharynx, or why do they behave in a different way? They all squamous cancers.
Yeah, so I would first talk about the base of the tongue or the oropharynx region. So there it's because of the transitional epithelium. It could be associated with HPV, and HPV alone could not drive, but it could be a synergistic effect between HPV oncoproteins and other mutations that would drive more, and that's why also it could like we have seen that HPV positive cancers are more they respond better to treatment. Apart from this, the cancers in tongue or the oral squamous cell carcinomas in tongue, they invade more into the muscles, whereas in gingiva, so they are more, even if they are smaller in size, they have a kind of an iceberg effect. So they are more into the bone, and if gingival carcinomas go into bone, then it's very poor prognosis. Apart from that, there are some studies comparing buccal mucosa and tongue also, but they don't see much of a significant difference in overall survival.
Yeah, but you are a stroma expert. I saw you have a nice lecture, and you studied the stroma. Do you think the stroma plays a role in difference in behavior? Did you see any clues suggesting further investigation of difference in the stroma? Because the stroma in the tongue should be different than on the gingiva.
Actually, yep, there is. Let me check table 1. Yeah. So we don't see significant differences in microRNA expression, at least that we have looked in oral squamous carcinoma based on tumor site like tongue or gingiva. But I propose, or I could hypothesize that the stroma in gingiva would be more suitable for proteomerogenesis, as it causes more bone invasion as compared to that in the tongue or in the buccal mucosa. So I am still, or I'm not a tumor stroma expert, but I could think of that.
That might be true, but just from a basic histological knowledge, you can see that the underlying stroma in the gingiva is different than in the tongue. Like you said, there's more muscle. But I don't know. Let's go back to squamous cell carcinoma of the vulva. It's more my topic. It's known that lichen sclerosis is related gives an increased risk for the development of cancer. The risk is not very great.
Yeah.
But do you think it's worthwhile looking at lichen sclerosis and the stroma and to make a model predicting which patient will develop cancer and which patient will not develop cancer? Do your data give a clue?
Yeah,
because
so
that's a big issue, because many gynecologists are treating lichen sclerosis as an important pre-malignant disease. It is difficult to treat.
yeah, so most of the studies that I have come across, they say that lichen sclerosis has a 3 to 7% risk of transformation to VSAC, but almost 30 to 60% of VSAC that is dependent is or has a background of VLS. And let's say if we just consider that it's just 3 to 7% increase, but still it amounts to some lives. And if we can, and we should save those people. So even if the risk is low, I think we should develop models to study that.
Yeah, but the question is, do you think the patients who develop cancer have a different form of lichen sclerosis than the patients who do not develop? So that it's due to the stroma that eventually a cancer develops, or is the event that takes place in the epithelium?
So there are not many studies that look into lichen sclerosis in particular, but then if you, I'm not a pathologist obviously, but if you look, I have looked at the stroma of lichen sclerosis, and it's almost nothing there, or it's very devoid of cells. And in addition, it has a immune some inflammation. So I would say it would be interesting to see what comes first. Like, is it because of the epithelia, or is it because of the stroma? So for now, I don't have a definite answer, but we are developing. We are in the process of developing a cohort that would look at VLS from past 20 years in our hospital, and we would like to look at what changes were there, or what percentage of VLS patients had carcinoma or progressed to cancer. So it would be interesting to look at it, but right now I don't have a specific answer.
No, neither have I, but it's always nice to discuss with an expert. So yeah, you are. So,
No.
Yeah. It's do you know the clinical situation in vulvar cancer? What are the problems? The basic problems. Do you have any idea about the history of the treatment of vulvar cancer? You have nice diagrams in your thesis, but
yeah, I think I know a bit, but it was first more radical vulvectomy, then it's now less radical, where they just take 1 centimeter from like tumor-free resection margin, and they also look at sentinel lymph nodes. To be honest, I'm not very sure about the surgical part or the treatment part, but I think still there is a stigma associated with cancer in that area. So that's kind of my knowledge in that field.
yeah, I see. The issue is, I think I'm old enough to see the whole spectrum. Let me tell you a small story. When I was a resident, 1 year, I saw a huge specimen with total vulvectomy with a very small squamous cell carcinoma of only 2 millimeters. And I had some knowledge of squamous cell carcinomas of the skin, because that was the first topic I had in my when I was studying for pathology. And I said, well, I would never do this. It's way too large. It's mutilating. And but that patient had 3 metastases and eventually died of the tumor.
Oh.
So that made me humble. Now we do, but the recurrence rate in that period, the local recurrence rate, not the lymph node metastasis, was close to 0. We did also do something good. Then we moved to smaller resections, and now we have a recurrence rate of 42%.
Yeah.
And even why is it important? Because you want to know, and I was wondering whether you have an idea how to solve that. How can we know which patient will show a local recurrence and which one not? Because any idea from your data, from your research?
So in our case also, we have almost 33% recurrence, and
it's a little bit lower, but your population, you have in your study population, there are quite a lot of small tumors.
Small cancers, yeah.
For us, it's the overall percentage, and that include obvious cancers growing into the anus, etc. Yeah.
So if we look at the resection margin, almost 75% are free of resection, and that leaves us like 25% with inconclusive or free. And if you compare that to the recurrence rate, so it's still more. So it suggests that the resection margin may or may not play a role in local recurrence at least. So I think there could be some biomarkers in the stroma that would guide this decision whether the resection margin plays an important role. But what I could see is, at least in microRNA 21, that it is much more expressed in even in hyperplastic tissue that is surrounding the cancer. And it would be interesting to look in a more bigger cohort how that affects local recurrence rate. Because for us, the recurrence was almost 33%, and that amounts to just 30 patients. So we could not deduce any significant interactions between microRNA 21 and local recurrence. But I guess it's a very important thing to do, at least in vulva cancer where the recurrence rate is so high.
Do you see, you look at the sections, do you see microRNA 21 in the resection margins? Have you ever looked for that, or?
We haven't looked at it, but what I understand from resection margin is the end of tumor that is either 1 mm or 2 mm, which does not contain the tumor. Am I right?
Yeah.
Yeah. So then, so most of our blocks have normal mucosa. Normal, I mean the mucosa that is not hyperplastic, or it has not it has not undergone carcinogenesis. In that, we don't see microRNA 21 at least. But still, I think we need to confirm this with special pathologists, because I am not one.
Okay, see. Because I always thought that the answer would lie in the epithelium. But you also write in your introduction the concept of field cancerization. And I think that most of the local recurrences are due to field cancerization and are de novo cancers. So it's not very useful, I think, but I may be wrong, to study the tumor you resected, but just to take biopsies from the other areas to see whether you like it sclerosis or DFIN or maybe stromal changes like you showed in tumors. Then the question is, do you think that in pre-malignant lesions you can already see stromal changes that will predict recurrence? So that's the main point that came up after reading your thesis.
Yeah, actually, we have a very good tissue biopsy repository here, and we would be looking into pre-malignant cases, both differentiated WIN, that is DWIN and UWIN, and look at biomarkers such as microRNA 21 if it can predict or if it is correlated with carcinogenesis or not.
Yeah, you were very optimistic about well-characterized precursor lesions. Okay, usual VIN is easy to recognize. I can teach you that in 5 minutes. But if you look at DVIN, you will see that pathologists will not always agree on that one. So we need a good biomarker for DFIN, and but that's still in development. And the whole concept of DFIN is, well, difficult. I'm not so sure whether it's reproducible. Yeah, you have DFINs that look like usual fins, and you have DFINs that almost look like normal hyperplastic epithelium. So I don't think morphology is going to give the answer there. But maybe, so that's why I like reading your thesis and think maybe I can find the answer there. So that's why. Do you? Okay, let's continue. How much time do I have? Can someone give me an answer to that? Hello?
I think we can discuss as long as we want.
As long as we want?
Maybe.
Maybe.
But I just wanted to ask, can I take a short bathroom break because I had too much coffee? Sorry.
Yes, of course.
I'll come back in 2 minutes. Sorry.
Yeah,
that's okay.
Yeah, thank you.
So,
how long does it take? How much time do we usually?
There is actually no specific upper time limit. I mean, the whole session should be finished like 3 hours or so. So, but normally the first opponent spends something between 30 minutes and 1 hour. So it's a little bit up to you guys.
Okay, so we are now on 30 minutes more or less.
Yeah, I've spent 30 minutes.
I could talk with him all afternoon, but that's not. So I just also for Else, I will stop at 1:30. Yeah.
Yeah, that's fine.
That's fine. Okay.
And I cannot see so far that there have been any persons opposed or wanting to propose to ask questions ex auditoria. So you have the time until the end of the first opponent to make yourself visible for that.
I'm so sorry, but I'm back.
That's okay, Harsh. These are the advantages of having a digital defense. So please, Harry, you may now continue.
Okay, thanks, Mr. Chairman. What about microRNAs? We can make a statistical association between behavior of tumor and the presence or absence of microRNAs. Yeah?
Yep.
What always puzzles me, what's regulating those microRNAs? Are they just a reaction or are they an initiator? What are we looking at when we are looking at microRNAs? Any idea? Because you can You can demonstrate there. You can do a nice in situ hybridization, very nice pictures, nice technique. But the question behind that is, what's causing the changes in the microRNA expression? Is that still the mutations in the tumor drivers, and then they are just a reaction, or are they causative?
So there are studies that show that they can be causative using microRNA mimics and inhibitors. We also have done some experiments with microRNA 204, 582, and which we show that when it is higher, like when we use mimics, it decreases the invasion in 3D models. But still, there are not conclusive results whether they themselves can cause, or whether it is a reaction of, let's say, genetic or epigenetic changes. So they could be drivers of oncogenesis, or they could be effect of mutations, as you said. They could be effect of mutations on expression of microRNAs. But still, it's still a growing field, this microRNAs, and more and more data should come up. Also, we will be studying some of the microRNAs in detail.
I agree. You can do also a microRNA array to study 250 or more even today microRNAs in a given tumor sample.
Actually, that's how we got our data also. So we did it in a microarray in OSCC, and we got some candidates. I guess 12 to 15 differentially regulated candidates. But again, it's very difficult to say if they are causing or they are the result of some other causes.
Mm-hmm. I asked, and it's not fair to ask you, I think, but our specialists in microRNAs do not have a clue why they are deregulated, those microRNAs. Do you have any idea? And if you do say, I don't know, that's quite fair, but I was just curious.
Yeah, so maybe then I should just say, I don't know, but I think, so they are, like in normal tissues, they are non-coding, so they don't code for any proteins, but they also regulate themselves. So they have, or whenever they are transcribed, they come from a exon or intron region, which we call it as junk DNA. But I think they can regulate themselves, or they can be regulated by other genetic or epigenetic changes. Right now, I don't think I'm very sure, but I see that it could be one of those reasons.
Yeah, I agree. I don't know, and the experts in my lab who work with microRNAs don't know the answer either. So it's fair enough. It's, but it's still intriguing. So it's also important, I think, if you do a microRNA assay and you find a lot of differences, that you do the in situ experiments. We also do usually the protein that is being interrupted by the microRNA expression. Do you also do that?
Yeah. So now we have just targeted calves in tumor stroma because we wanted to study that. But next we will be combining this. So we'll combine both the microRNA that is differentially regulated and its downstream target or upstream target. And then we might know better because this method was established to look at that. And maybe I will continue working on that in future.
Yeah. Well, then the pathologists are still useful. We can say in which cell does express the microRNA. Yeah, if you just make a soup out of it, it's somewhere in the tissue, but you don't know what cell. So that's good for the future of pathologists.
I think pathologists will always be there.
Okay, thanks. Well, I'm almost retired, but I'm a little bit worried about my residents. I would like to have a small discussion about P53 in vulvar cancer. You quantitated that. Why?
Sorry?
You quantitated the expression of P53. I think I would never do that. You know why? Because I think the pattern is more important than the number of positive cells.
Okay.
There are some nice recent papers that describe the 5 patterns in squamous cell carcinoma, but
Um, just to be here,
yeah,
actually here. Yeah.
yeah.
So I would just like to add something. So we also see 3 different patterns. So one is that have less than 1% of p53 positive, and then some are more than 50% positive. So even less than p53, 1% p53 could indicate a mutation, and more than 50% of p53 could indicate a mutation. So based on this, actually I did some Cox regression and I did not find any significant differences. So for this, I've just presented it based on median cutoff. But yes, I've gone through some papers which suggest that no p53 is also an indication of mutation.
You know, if you have a highly characterizing carcinoma, most of the tumor cells are on their way to apoptosis, and only in the basal cell layer contains vital cells. And you can have a very low number of p53 strongly positive nuclei, and still, but if they fall like p16, if they are blockwise, they are present, and more than 95% of your basal cell layer is positive, then it's a mutation. So that's why I say I don't count them. I look at the pattern. So in poorly differentiated cancers, you may have 80% or 90% p53 positive tumors, but that's why I don't like. Key 67 is a continuous variable that needs no interpretation. But and then that one you can count. The p53, I would just look at it. But
so you would. I'm just trying to understand, like with the pathologist view, how would you like to describe
I would say
it?
Positive or negative.
Okay, so just positive and negative based on the staining.
Positive and negative. There are several. You have the 0 phenotype with no protein that is correlates strongly with a p53 mutation. You have the blockwise strong staining that is a mutation. You have a very rare mutation that gives rise to accumulation in the cytoplasm. It's far less than 1%, and all the others are wild type. And if you follow that up with a gene analysis, that is correlates for more than 95%, not 100%. It's never 100%. But that's how I would look at p53.
Okay. So maybe we will, or I will try to improve this.
Yeah.
And mention it. Yeah. Thank you.
That's just a small comment. Well, I'm out of time, I think. It's nice for Else has focused on paper 3 and 4. So she's not a pathologist, so I think I do the pathologist things. And she can harass you with difficult
if there is something
on that number 3 and 4. So I close the microphone.
Thank you, Harry, for a good discussion.
Okay, so then I thank the first opponent, Professor Harry Hollema, for his opposition. And as I'm not registered any person who has requested to speak ex auditorio, we are ready to move on to the second opponent. However, I was advised from the administration that it's good to now put in a 5 minutes break, so people can maybe move a little bit around and so on. So I think we will do that, and we will start exactly 13:35. So if the candidate and the second opponent and the others are ready in 5-6 minutes, that would be excellent. Thank you.
Yes.
Thank you.
So then I think we are ready to start again. And that
Sorry.
the second opponent is with us, and it's then a pleasure for me to hand over the word to the second opponent, Else Driehaus. Please.
Yeah, thank you. So Harsh, first of all, of course, I'd like to congratulate you on the nice thesis that it was a pleasure to read. And I'd also like to congratulate your promoter and supervisors.
Yeah.
I'm honored to be an opponent today, and it was a pleasure to read on your thesis. First of all, the double or even triple staining protocol, if I understand correctly, you developed for the in situ combined with the immunohistochemistry, and in particular the applications you used it. Mostly for me, it was very interesting to learn more on the interaction between the stroma and the epithelial components of the tumors. And with my background, as you may not be surprised, for me, it was particularly a pleasure to read on the in vitro models that you've applied to study the interaction between stroma and epithelium. So this will also be the focus of the questions that I will ask you. So I will mainly focus on the paper 3 and 4 of your thesis. Yep. And I also have some basic questions on the methodologies, but I would also am looking forward to discuss with you the work that you've done in a somewhat broader perspective, actually. So I've prepared a PowerPoint to maybe help the discussion. So I will share my screen.
Yep, I will stop sharing mine. Yep.
So let's see. So you can see the PowerPoint right now.
Yeah.
Good. Yeah, so I just copied some of the figures from your thesis, which I had in a PDF, to thought that it might help the discussion. So I just, before we move to paper 3 and 4, I have some quick questions on mainly the implications of the work that you've presented in paper 1, where you, if I understand correctly, so please correct me if I'm wrong, the main conclusion that I drew from reading paper 1 was that the expression of microRNA 204 in the fibroblasts or cancer-associated fibroblasts is associated with a better prognosis or overall survival in the patients with the oral squamous cell carcinoma.
Yeah, just to correct you, it was in stromal cells. So yeah, we did not use double staining, so we don't know if it's in calf, but it was in stroma. Yeah.
Yeah. Okay. Yeah. And then I read in your introduction that there were also others already that found this specific correlation between expression of this particular microRNA in the tumor as a whole, predictive already for outcome. So now you add to this, because in addition to establishing, of course, this very nice staining model, which I think has many applications, it allows you to discriminate in the fibroblasts that are positive for this marker rather than just bulk tumor tissue having a positive staining. But what would this add if what would the detection of a fibroblast specific expression have a benefit over just the bulk positive expression of microRNA in the tumor?
Yep. So I think this would just add in selecting a cohort or subset of patients that overexpress this microRNA specifically in fibroblast, and then we could target this overexpressing microRNA fibroblast in some clinical studies. Then it would add to the treatment. So basically, as is same with most of the immunohistochemistry methods that are on pathology based, they add in differentiating between patients which might have a better prediction or which might have a better or favorable response to certain therapies. So expression of this microRNAs in fibroblast, it could help in selection of patients that would, that's just as I said, that it would help in predicting survival or predicting some therapy that could be used.
And then for my basic, because I have limited knowledge on this topic, so you show now that this microRNA is actually expressed in the fibroblast. So then it got me to think, because you mentioned in the introduction of your thesis actually that there are some studies that claim some potential targets of these microRNAs, right? To target then the RNA encoding oncogenes or tumor suppressor genes, making the microRNAs rather oncomers or this tumor suppressor miRNAs. So then, are the molecular targets of this particular microRNA known?
Yes, actually it is known to target integrin alpha 11, which is a marker of CAFs, or it is still in progress, like the research is going on. And also not directly, but it has shown to affect FAK, that is for focal adhesion kinases that are upregulated in CAFs.
Okay, so the idea is, because that was also what I was thinking about, and I wanted to get your opinion. Do you think that the microRNAs that are expressed in the fibroblasts, that they mainly act intracellularly, so they stay? Is it autocrine signaling? So is it do they affect expression? Well, maybe signaling is not the correct term here, but do they affect expression in the fibroblast or in the epithelial component of the tumor? Do you think that they're actually microRNAs reaching actually the epithelial cells, thereby deregulating the pathways in the epithelial cells? Is there any knowledge on that?
Yeah, so there are studies that have looked at exosomes or the cargo in exosomes, and they have shown to have a different microRNAs. So I think there was this microRNA 34 that was upregulated in some form of cancer. So I don't know which form of cancer, but it has been used in therapeutics as well. Of course, it failed, but yeah, there are some cargos. So the exosome cargos could, in a way, act in a paracrine way. Apart from this, there are also studies that suggest, as in case with other growth factors, that these microRNAs that are secreted can also act in a autocrine fashion and then carry out its target signaling.
Okay, thanks. So then this method of combining in situ and immunohistochemistry in the same slide, actually, I think you explained very well in your thesis what advantages this has over doing stainings on separate sections, where you, of course, are always on a different level in the tissue. So when you think about you having developed now this technology, then if you work either, well, you work in research or you would work in the clinic, and you have now this new cool tool in your toolbox, what can you do? What does it add currently, or what can it add in the future, either to basic research or to the clinic? And what are, for example, also its limitations?
Yeah, so I think we briefly, me and Harry, we briefly discussed its implications. So in basic research, it could we could use this method to look at the direct microRNA targets, and then we would see in a single section what microRNA or how it affects downstream signaling or upstream signaling based on protein expression. And once we have some microRNAs that are very specific or that are clinically significant, we could implement this in a clinical setting as well and look at both the microRNA and its downstream targeting, so which could also help in determining its clinical benefit. So talking about the limitations right now, so the protocol is based on protease degradation of the tissue. So some epitopes, they don't work with protease degradation. For example, I think one of the cytokeratin that I used did not work, but pan-cytokeratin, that stains all type of cytokeratins, as well as alpha SMA, it worked with this degradation. And apart from that, some of the epitopes, they might be degraded during hybridization. So the hybridization it's microRNA dependent, but it's mostly about above 48 or 49 degrees. So some of epitopes could be degraded by that way. But if we look at just plain basic immunohistochemistry, that involves boiling it at 95 degrees to expose the epitopes. So that should be fine. But the in situ hybridization buffer has formic acid as one of the component, so it could also destroy the epitope. Apart from this, I think it's a very sensitive technique. So most of the protocols that have been established are for fish, that is fluorescent in situ hybridization, and they are more sensitive because you can just close one channel and see. But then they do not combine the histology of the section. That's why we think chromogenic are, at least we think that they are better.
Yeah, thanks. Because I was thinking about actually comparing this new tool that you developed with other new technologies that are used. One of them being, for example, I put an example here, a publication in Cell on the single cell RNA sequencing of actually primary head and neck cancers, which I don't know if you know this paper, but particularly I think it might be of interest for you because they do single cell seq on these tumors where they also find fibroblasts, obviously, of course, in the tumors. And actually further along in the paper describe two different subsets of fibroblasts, which then they suspect might have different functions. And then, of course, I was thinking comparing what your technology allows is seeing the expression of microRNAs or RNA protein or RNA molecules, and at the same time keeping histology-based stainings to see in which cell type particular RNA molecules are expressed at that time. So then, what does that help you over, for example, a technology as single-cell sequencing, where you can see RNA without being selective, for example, by having to use an antibody or a probe? You look unbiased at expression patterns in all cell types in such a tumor with single-cell seq, for example. So what can your technique do that, for example, this one cannot do, or how could they maybe complement each other? Do you have ideas on this?
Yeah, so with single-cell RNA-seq, I think there are two ways. So one is it might not capture the whole heterogeneity spectrum if you just look at single cells. Another one is the location. So if you do a single cell sequencing, you will not know whether it's close to the tumor, in the tumor center, tumor front, or far away from tumor. You'll get same, or you might get some inconclusive results. But what this technique has is the location. So if it's at the tumor front, you will see a difference expression. Maybe if it's in the tumor center, it will have different possibilities. So this technique would add more to this. So I'm not saying it's the only technique, but it would act as a complementary technique to other techniques available.
Yeah, I agree. I think your technique, indeed, the location of the cells, which is of course a very important one, especially you actually also mentioned differences between tumor front and tumor core, right? Is something that is lost in technologies such as these. On the other hand, this technology, as you say, can do maybe this is a more unbiased approach, right? So you're not limited to what you stain for. So I was thinking maybe it would actually be good to look at their expression in the fibroblasts that they find to see if actually they detect the molecules that you studied. And with that, I'd like to move on to actually paper 3. With some basic questions, basically. So I copied here a part of your methods where I read that something you actually also mentioned in the presentation preceding this discussion, that out of the primary cancer cell lines that you could isolate, which were 4, one could be propagated indefinitely. So this is the VCC1 that you then move on to work with. So what I was curious, what do you define as success? Because you did say that you established 4 primary cell lines. So then did they stop after, how long could you grow them?
So like I'll start with the all 7 tissues. So out of 7, we just could isolate 4. So out of 4, the other 3 could be propagated up till passage 3 or 4. But then in order to grow them further, we need, or I immortalize them using E6, E7 vectors. And they could not be grown indefinitely without implementing any additional genetic alterations. But this cell line VCC1, even without transforming, it could grow. Now it is in, I think it's almost in passage 75. So it's more than 200 population doublings. So this was spontaneously, continuously growing cell line. So, but then talking about the differences, what I think is the turnover of vulvar keratinocytes is different from, let's say, oral keratinocytes. So especially in old patients, I think, I'm not sure, and actually I don't have any scientific data to support this, but I think the turnover in oral keratinocytes is more because of the continuous chewing and other mechanical forces, whereas in vulva cancer it's not. And that's what we also see. So when we isolate them, they grow for 3-4 passages, but then they just die automatically.
Okay. And there was no particular characteristic different of this one sample out of the 7. I know they're small numbers, but you kind of sometimes have this feeling when you work in the lab with stuff that that made this one different. So do you have an idea why this one kept growing and the other 6 did not?
Yeah, so to be honest, this was the first tissue that I isolated, and maybe I put more effort in it, but I'm not sure. But scientifically, I don't think there were differences. In some tissues, I guess 5th and 7th, the tissue was small, so we did not have that much tumor part.
Yeah.
And apart from this, I just want to say that so this tumors are very keratinized, so they're very hard. So when I cut them, I could feel that it's all just keratin, and in sometimes I could predict that maybe the cells will not come because there is nothing left, or I don't know, but that could be one of the reason. And yeah, as I said, this was the first tissue that I isolated, so maybe I put more effort.
Yeah.
So
yeah, because I also noticed in the methods, if I got it correctly from the methods, that for the wild type tissue or tumor adjacent wild type, you actually digested the tissue and then separated the epithelial layer from the rest, and then continued digestion to make cell suspension. But for the tumor tissue, you cut it up in pieces and then plated it on plastic. Why this different approach? Why not digest the tumor tissue or also cut up only the wild type tissue?
Yep. So in wild type tissue, we digest it with dispase. So dispase is a collagen 4 degrading enzyme. So it degrades the basement membrane. In normal tissues, the basement membrane is well defined actually. So you have difference in the epithelia and stroma, and it would degrade. So you can just separate it out. But in case of tumors, there is no well demarcated basement membrane. So actually, you can't divide it. And so my supervisor is working in this field for 15 years, and she has seen that when there is no additional effect of digestion on tumor tissues. So this is kind of a well-established protocol in our lab, and that's why we don't isolate tumor tissues. But that said, it could also be because of this method, there could be some difficulties in replicating the same effect, because these are 2 different techniques. So in order to rectify that, what we are doing is we are now growing, or we take the wild type tissue, cut it into 2. One goes into dispase digestion, another one we just perform it or follow the protocol same as for tumor tissue. So we don't get any selective population there.
Yeah, because I also noticed that you separate then in this process the epithelial components from the fibroblasts, because you want to generate the different cell lines, which you then later on combine again, right, in your cultures. But the main message conveyed in your thesis is actually that the stromal the interaction with stromal cells might be very important for a tumor or the epithelial component of the tumor to be able to proliferate or grow or migrate. So do you think actually the separation of the components in the beginning of your methods could affect your success rates? And have you considered not separating them and then seeing if your epithelial component can be grown better?
Yeah, so I think, so when we separate them, as I said in my trial lecture, that when you separate them, and even they are, let's say they are normal fibroblasts, but once they are in culture plates separated, they become activated, and they might definitely behave differently from the normal fibroblast. So yes, it's a good point. And for now, we have not used this, But for the past 6 months, what we are trying to do is when we collect the tissue samples, we grow them on collagen, collagen type 1. So then it has not seen the culture plastic dish, so they are not kind of activated. We don't know yet how we'll play with the stroma, but we have started to do that. And right now, I don't have a definite answer if it affects, but maybe if we talk after a couple of years, I might be able to answer it better.
Yeah, it would be interesting, right, to see if this might actually help your establishment of epithelial cells in culture.
Especially, I guess, in carcinogenesis, that it might play a different role. Yeah.
And then you will not be surprised that I was interested by this figure, which you mentioned. You call these structures spheres. And they resemble something that I call organoids. And then you mentioned when you compare your newly established VCC1 cell line and their sphere formation of that cell line with the other ones that were already established previously, that there was a similar amount of spheres to this UMSCV1A, but then overall spheres were larger in size. And then I was just curious. I mean, I understand that you compare it to already existing models. What do you think it says about cell behavior if a sphere can be formed? That's one thing. And then secondly, the size of the sphere, because I was thinking about it and I found it hard to translate this to, I don't know, a biological meaning, but you do mention it in your paper. So I was wondering if you have an opinion on this.
So, so what growing spheres means that it can grow independent of the contacts in cell culture plates, so which suggests that they have more stem-like features. And so if they are bigger, then it suggests that even though there is some hypoxia in the center, in the core, the surrounding cells, or they kind of cover it up, or they balance it out. So bigger tumors, bigger spheres, I would think that it is due to increased stemness of the cell lines.
And then the number of spheres, or the sphere formation capacity, does that hint to the same thing, or is that something different?
Yeah, so actually we start with 500 cells in each well. We also tried with 250 cells. So this also reflects on its clonogenicity, so that it could form clones of the same cell type or of the single cell. And I think the number of spheres and number of clones directly relate to the stemness, or at least in cultures, it behaves like more stem-like.
Okay, so it's a bit similar to the size in your opinion, right? So then I was curious because these structures that you call spheres remind me of what when I was working in the lab of Hans Klevers, I called organoids. So have you, just out of curiosity, have you compared the behavior of these epithelial cells in these spheres to how they behave in 2D cell cultures? And more specifically, also, for example, when you go back to the beginning where your success rate was 1 out of 7 for the long-term culture of these cells, have you considered trying a 3D model, or do you think it makes sense?
Yeah. So coming back to your second point, that is, have you compared 3D spheres to 2D cell cultures? Yeah, actually we did. And so all these methods that I've done in paper 4 were actually on a 2D culture plate. But then we could see a very distinct difference when we grew them on Matrigel. It could be also that Matrigel played a part, but the 3D spheres are definitely different in cell cultures. Also, I looked at this PI3K AKT pathway, and they express differently in 3D spheres where they are activated, whereas in 2D cultures, yeah, I don't see that much difference. And then coming to the next point, that is trying 3D spheres establishment from primary tissue. We have not tried it. We have not tried it yet, but I guess it makes sense to directly make organoids or spheroids from primary tissue. So maybe in future we would cut it into half and try one of this method and try one of our methods that are already established.
Do you have an idea why your cells might behave differently in 3D than in 2D?
It's again with the signaling. So in 3-dimensional, they have, let's say if they're in collagen gel, so then they have different epitopes, different integrins that are activated, and it might play a role in outside to in signaling. Whereas in 2D, they are connected. They're just on the plastic culture. They have to secrete their own matrix, and which which will affect the signaling.
So do you think the matrigel or the other extracellular matrix you use when you start growing organoids or spheres could take away part of the effect that is required in 2D by adding your stromal cells?
I think the signaling would be different anyways. If you look at matrigel, it's already isolated from a tumor sarcoma, I guess. Whereas in collagen, it's non-activated, but the receptors that are for binding to the matrigel or to the collagen are different in stromal cells, and it will play a very important role actually in the signaling, and also with the paracrine signaling involving the cancer cells or epithelial cells.
Yeah, so you expect indeed that basically repeating the same experiments might give different results if you do it in 3D in matrigel, for example.
Yeah, okay. Actually, yeah, that's what we did in some of the experiments in paper 4, and we could see very different results. So then we switched on to matrigel. So I acknowledge that it's not the best matrix, but we would try again with collagen also, and different concentrations of collagen, fibronectin, and tennessine maybe. Yeah.
Well, I'm not saying it's not the best, or but it's interesting also for me to hear that, if you choose an in vitro model, there are differences, and you could actually get very different results. So it's I think indeed, as you say, it's important to compare and to realize what you change. And I think actually you knowing a lot more about tumor stroma than, for example, me, the change to collagen from matrigel, for example, says something much more to you than it does to me. But it's definitely good to hear that you're exploring this, and I'd like to hear indeed in the future what came out of these. Then you use this method. So I copied this again, of course, from the publication to confirm that there was no cell swap, basically, right? So that the cell line was indeed originating from the donor. Exactly. But as far as I could find in paper 3, but maybe I missed it, you didn't do any confirmation that actually what you're growing is tumor, the epithelial cells. Are you confident that you're growing tumor? Are you not growing wild type cells? Why are you confident that you do? Are there ways to test this?
So you're saying that the cells that are isolated are tumor cells indeed?
Yeah, have you confirmed this?
Yeah, so actually we did NGS also.
Okay.
In between the tumor donor tumor tissue and the cell line, and it showed similar actually mutations. So we are kind of confident that, but I don't think I have included it in the paper. So
no, I was just curious because indeed the next question was then if you did NGS, then you have this answer. What I'm always worried about when I establish 3D cultures from primary patient tissue is that, or what I try to check for, is that you can imagine, if you transfer it to an in vitro environment, basically, you might select for a specific tumor clone, maybe the one that is most aggressive, maybe not. But whenever you pick up this model to then say something about the clinical, for example, the patient progression or the treatment of the patient, you might be off because the model you have in the lab is actually only part of the tumor in the patient. So now you told me that you actually did NGS. Did you see that maybe you selected for a specific clone of the tumor of the patient, or was it recapitulated as a whole?
Yeah, so what we saw, the mutations in NGS were similar to the ones that the patient carried. This was done on, I guess, after 15th passage. And we also did it again on 38th. And we could see it was same. but now it's been growing over 70%, and it would be actually a good idea to see if it's still recapitulating. But I think from my experience, I would suggest that it has acquired more mutations and become more selective.
Okay.
So that's why if I had to do more experiments with this cell line, I would choose the earlier passage, which are more similar to tumor.
Yeah. Okay. Thanks. Then you move on to actually combine the isolated fibroblast with the epithelial compartment of this tumor, right? The cell line. And then I took it from this figure that the invasion was enhanced by the invasion of the epithelial cells into the downward was enhanced by the presence of stromal cells, although the origin of the stromal cells, so normal tumor, didn't matter in this case, right?
Yeah.
So then is there really such a thing as intrinsically reprogrammed tumor stroma, or do you think the epithelial cells actually instruct the stroma, or because you get the same result whether you use normal or tumor-derived fibroblasts here, right?
Yeah. So what I think is even when you isolate normal fibroblasts and you put them in plastic culture, they become activated, and they act as like they are transformed, or they are activated. And then irrespective of the presence of cancer cells, let's say VCC1, they behave similarly. But we have also done some experiment with oral cancer cells, and in that it is different. So we are not sure if the oral normal fibroblasts are special, or if the vulvar normal fibroblasts are activated all the time.
So what is different? So you don't see activation by the normal fibroblasts in oral, or what is the difference?
Actually, I wouldn't put it the complete activation, but it reduces invasion in oral fibroblast.
Okay.
And so it was not like I did not study, and it was not part of but we see two different phenotypes again in normal calves and oral calves, and that subtype could actually be lost in vitro culture. So if you use passage 3 or 5, they might have been lost. So all these experiments were even performed in almost similar passage, same passages, 3 to 5.
Mm-hmm.
But my thinking is that those that are more activated survive that passages, and others that are not, they actually die in P1 or P2. So I think your point that we should use directly the spheroids to generate this might have better, or might be more clinically relevant.
Yeah, I don't know. It's worth to try, right? And to compare. I don't know what is better. And then I'll skip this for sake of time. Yeah, could you think of I was thinking I miss a control. I don't know if it's something that is actually possible in the model that you have.
Mm-hmm.
But can you think of a control in this image particularly that is missing?
Yep. So the control could be a cross-species fibroblast. So we tried with mouse fibroblast to see if there is a cross-species reactivity and whether they secrete the same growth factors. But to be honest, because of the lockdown, I could not see the results, but it's there. Maybe in a couple of months, I would let you know.
I was thinking about the control where you have wild type or healthy epithelium on top, because that might help you address the question if the stroma is actually also not activated because of the in vitro model, but if the tumor epithelium is instructing it in some way.
Oh yeah, sorry. In that way, we have isolated normal keratinocytes, and using the same fibroblast, we have seen that it does not invade. So it's it looks like the cancer cells are activating the fibroblast to secrete MMPs or to degrade the matrix.
Yeah, or it could also be right. It could also be that the fibroblasts in your conditions where you have the normal epithelial cells with the fibroblasts still do the same, but the normal epithelial cells do not respond to it. Right? They're not activated and they don't. Okay, but it's good to know that you've done these comparisons. And then I was puzzled by this particular finding that this in the top the same figure we just looked at in paper 3, and then in paper 4 you do a similar experiment, as far as I understand. And there you actually do see that only the tumor or the fibroblast isolated from the tumor result in tumor migration. What is the difference? Do you have an idea?
It's the cell line actually. So UMSCE4 is known to be more non-tumorigenic, so it does not form tumors in nude mice, whereas VCC does, and they are known to be inherently non-tumorigenic. So then it relies much more on the calves to secrete some signaling molecules that would help them.
So that would mean, following this rationale, that would mean that there is a difference in vitro between the normal fibroblasts and the cancer-associated fibroblasts.
Yes, actually, but it could also be more of a synergistic effect between the cancer cells that are growing on top and the fibroblasts that are in the stroma.
Yeah, it would be interesting to understand these differences better, right? Because this is particularly what might cause the differences in clinical behavior we see in patients that present with, on histology comparable tumors. And then a bit broader. So I think your data shows that the presence of, I put here, cancer-associated fibroblasts, maybe I should say fibroblasts, in a model system affects the tumor cell behavior in vitro, right? I think you have shown very nice co-culture data or models here in this paper already that allow you to investigate the interaction between stroma and epithelial cells. And with your transwells, you can actually do it both ways. So in your opinion, what does this imply for actually, basically, there's now paper after paper coming out actually that show that organoids or 3D models established from patients actually predict clinical responses of the patients. So I put just 3 here. So these are, I think, all actually on colorectals, but we also have them. They're also out for other. They're out for breast, for head and neck, for pancreas. That actually the organoids, which then in these cases only consist of epithelial components, predict patient responses to therapy. There's no stroma in these models. Do you think we miss, if you do it in such a way, you miss a subset of patients? Do you think, what's your opinion on this? Because I'm convinced after reading your thesis that the stroma is an important thing to take into account in vitro.
So what I think is these models that predict or that predict response, like chemotherapeutic response, are only associated with a subset of patient that in which the cancer is driven by the epithelial or the epithelial cells or just the cancer cells, and it does not take into account the stroma. So maybe if we have something that mimics both, we might be able to catch a larger subset of patients that respond to this or does not respond to this.
So these papers already present a correlation already between the lab and the patient. So for at least a part of the patients, they can already do it and they don't have the stroma. So maybe I don't get your answer.
What was the correlation like? Was it 50%, 80%?
Generally, it was between 80 and 90% actually. But actually, the one that is in the bottom in blue, that actually is an interesting one. So that might actually address the if I understood your comment correctly, there was a proportion of patients they missed. And I spoke recently to a researcher that thought it was because the immune response was essential or the immune component was essential to predict the response to radiotherapy. But then I heard you in your talk this morning that you say that actually stroma might also affect. But definitely, I think this suggests that it's not always required to have stromal cells in culture to say something about the response of the patients.
Yeah, it depends on, actually, I would put it like this, that it depends on the stroma. If it's very dense and then if it obstructs some of the chemotherapy reagents from reaching the cells, then it will definitely have a say. If it does not, then these 3D models are excellent models that can recapitulate what is happening in the clinics.
Yeah. Okay. Yeah, I agree.
Elsa, just a second. Is it okay if I take one more small break?
Yeah, sure.
I had too much coffee. Sorry.
Okay. So there will be a little break.
Sorry, I'm back, and maybe I should not drink more water.
Yeah. So thank you. So then I think that the discussion can resume. Please go on. Was it your turn, Elsa?
Yeah. Yeah. So then in this paper 3, you actually basically present a model to validate the findings in paper 2, right?
Yep.
Oh, sorry. Let me switch back to, yeah. And then in paper 2, you describe observations actually already on the microRNAs, and that actually expression of microRNAs in stromal cells can predict patient outcomes. So can actually the model described in paper 3 be used to validate or better understand this correlation that you see in patient data? Because there you always wonder if it's a causative. That's actually also what Harry also mentioned, whether it's causative or it's an association. So can you use your model that you now present in paper 3 to study this, and then how?
Yeah, so we can use the models presented in paper 3, and we have done some experiments with mimics and inhibitors of microRNA to see if it increases invasion or whether it's a causative and association. But these are still in preliminary, or it's still preliminary data, and we need further repetitions of the experiment.
So do you see expression actually of your microRNA 21 in your isolated fibroblasts in vitro?
Yeah, actually I have a presentation, but yeah, we see in vulvar calves that microRNA 21 is more expressed than the normal calves, as well as the normal that is surrounding the cancer, so the juxtatumoral also.
Okay, interesting.
But yeah, but so as we have seen in paper 2, that even though we see more microRNA in vitro test in V calves, it correlates with better survival. So actually we need further studies into that.
Thanks. Then shortly only some a smaller amount of questions on paper 4, where you use a cell line where you overexpress E6 and E7 as basically an in vitro model to mimic HPV infection, and then see how this affects your interaction with fibroblasts or cancer-associated fibroblasts. So basically, I was a bit puzzled by the first part of the results, where you start off with 2 cell lines, your VCC1 and then this UMSCV4. And then it says, in light of these findings, so I copied it here, further experiments were conducted only in this particular cell line, because you saw more effects, for example, as you mentioned, on YAP expression or phosphorylation.
Yeah.
And then in your discussion, you come back to this and you say that in this particular cell line, you saw more an effect of your overexpression of E6 and E7 on the cell cycle. So basically, you saw a bigger phenotype of your overexpression, and you chose to only continue with the cell line.
Yeah.
And you say, in light of these findings. So I wanted to ask you, what does in light of these findings mean? Because why wouldn't you continue with the other one as well?
Yeah. So maybe I'll just share my presentation. Actually, we have used VCC1 also. So if you can see, this is the VCC1 data. And using gene expression using similar models, we have seen that it also increases PDGFR beta and VGFC only in E6, E7 co-cultures. But we did not see any effects in vivo tumorigenicity. That is, they did not form bigger tumors in E6, E7 cells. So we have used this, but considering more phenotypic changes in UMSCB4, which had a T53 mutant, we used or we just focused in that way. But obviously, when we will move on to, or when we go to publications, we would like to include this data as well.
Yeah. Okay, thanks. Yeah, I agree with that, because of course your findings are very interesting, but in the clinic you also have patients with different phenotypes, and just picking one because it shows the biggest effect, you actually need to see what if HPV infection changes the interaction in the bulk of the population, right? Or in the bulk of the patient.
Yeah, obviously.
But then I'm happy to see this slide where you still see the effect. So that's very encouraging, I think.
But but we don't see the same effect in vivo tumorigenicity. But
So do you have any idea why?
It could be the intrinsic properties of VCC1 cell line, maybe. Yeah. So the E67 might not have, or it might just have an additive effect and not a synergistic effect in case of E67, whereas in UMSA4 it might be a synergistic rather than an additive effect. But we need a lot more experiments to prove this. So.
Yeah, I didn't add this to my questions actually now, also for sake of time. But I was already. I'm not saying it is not a synergistic effect you observed between the E6, E7 overexpression and the interaction with your with the fibroblasts. But I think indeed this is the thing that in the discussion you mentioned it's synergy, and I'm not 100% sure I'm convinced about whether it's additive effect or synergy. But that's something maybe discuss better later. So then I'll quickly move on to the final questions that I have. So I think you can see my screen again, right? So just some final questions. You overexpress E6 and E7 as basically a surrogate for HPV infection in the clinic. Why haven't you tried HPV infection just with actual viral particles?
So
it's a good idea.
So this packaging of HPV viral particles is depend on the differentiation, and which is observed in 3D organotypic models. So we have collected the condition medium from the organotypics, and we would be looking into the effect of HPV viral particles, because I definitely think that the infection with viral particles will have a different phenotypic than the E6, E7, and it will add to the paper quality of the paper.
Why do you think it would have a different effect?
So increasing E6, E7 oncoproteins will maybe directly change the phenotype, whereas if you add HPV viral particles, then it has to bind to some receptors on the cancer cells, and this activation through receptors might have a different role in signaling, which might not be similar in case of E6, E7 oncogenes.
Yeah, I agree. Then, interestingly, I found I'm definitely not an expert on FCCs, but I found that HPV positive FCCC actually generally has an better overall survival rate. How does your data fit with this, where you actually show that E6, E7 activates this, or well, aggravates or maybe even synergizes with the interaction between stroma and tumor cells?
Yeah, so I think most of this better survival is because of the response to therapy rather than their own effects. So HPV phenotypes are, even in oropharynx or in oral cancer, they are known to have a better prediction or better correlation with therapy.
Yeah.
I am not sure yet exactly how this works, but that's what I have read in most of the papers.
Yeah, I agree with you. Would be interesting if you, because actually a lab model where you have HPV yes, HPV no in otherwise the same cells is actually a very good opportunity maybe to actually investigate this, because
Yeah.
I don't know if it's understood well why this is the case, so why HPV positive tumor patients with HPV positive tumors actually do better. It's also believed they have a completely different etiology, right?
Yeah.
So if you have the same model where you could add or withdraw HPV, you actually can test this hypothesis, because there you would have the same etiology and then add the virus on top.
Yeah. Is it okay if I write this down so I can maybe work on it?
Yeah, sure.
Yeah, thank you.
So last question before I move to my final slide. Assuming it's synergy between the interaction between stromal and epithelial cells that makes the epithelial cells basically more invasive and more helps with the lymph vessel formation, together with the infection, in this case with or overexpression of E67, assuming that is synergy. Would you have an hypothesis on the interaction between this HPV infection and your fibroblast? Why do they synergize? How would it work? Do you have a molecular concept behind this? Do you have an hypothesis of this?
So I would say it's a very bold hypothesis, but what I feel is in HPV, you have even for like T1 and T2 tumor stage, you have a lymph node metastasis. Especially in vulva cancer, correct me if I'm wrong, Harry, but you see very less distant metastasis, so you see less than 10%, whereas you see almost 30 to 40% lymph node metastasis. So which could be an inherent property of VSCC cells, like they metastasize to lymph nodes. We don't know yet. But again, in HPV, just focusing on HPV, we have seen in oropharynx as well that even for T1, T2 stage, you have a lot of lymph node positive, or at least N1, N2 positive. So it could be that the interaction between HPV's positive cancer cells and CAFs could drive this pro-lymphangiogenetic response. And maybe if we target this synergy, or if we target this pathway, we could increase, or we could even reduce we could increase the survival rate in HPV positive cancers.
Okay, thanks. Final question, which I always I think it's a nice one to end with. So based on everything you mentioned in your thesis, which I think is actually very nice, that you have both data that is obtained from patient samples, so clinical FFPE data, and then moving into the lab to have models built to validate the findings you've done in the clinical or in the patient samples. If you would continue on this work and you would have unlimited funding, so no problem there.
Yay.
How would you proceed then, taking the knowledge you've obtained and you present in your thesis to aid the treatment, diagnosis, or whatever to part of the patient treatments to improve the outcome or the treatment?
Yeah. So first of all, so I would just tell what I'm doing right now. So what we are doing is we are establishing in situ hybridization for HPV, that is E67 dependent, and combining it with P16 immunohistochemistry. So that would help in selecting subset of HPV positive patients, because if you just take in situ hybridization or just the DNA testing or just the P16 immunohistochemistry, it does not correspond to 100%. So maybe if we combine both these methods, we will be able to select a subset of patients that would have better prognosis in terms of HPV positive. And this could be used in both OSCC, even though OSCC has very less HPV associated cancers. So we will be targeting more on head and neck that has the oropharynx as well, and it could target the HPV. And in VSCC, which has, it depends on what definition you have for HPV, but it is like almost 30%. So that would be one of the things if I have unlimited funding. The next would be to look at this microRNA mimics and inhibitors, whether it's a causative effect or it's based on some genetic or epigenetic changes, and how it could be translated to clinics using some therapeutics. And yeah, the last one would be to use or to base this HPV association with lymphangiogenic or lymphangiogenetic response, and how targeting CAFs would maybe prevent this lymphatic spread.
Well, thanks very much. Sounds like you thought it well through. Thanks a lot for the very nice discussion, and that were my questions.
Thank you.
Okay, so then I would also like to thank the second opponent for a very interesting and thoughtful discussion. And the committee will now withdraw to our digital discussion room to evaluate the public defense. The committee will then return to announce the result, and we ask the audience to remain online for this announcement. Thank you.
Harry? Harry? I think you should be in the other room.
So, Harsh, can you hear me? Yeah. The evaluation committee has evaluated defense of the thesis as satisfactory, and they recommend that the candidate is conferred with a degree Philosophia Doctor at the University of Bergen. The candidate may now express his thanks. Do you want to say something? You need to turn your microphone on. Turn on your.
Sorry.
Okay.
Yeah. So yeah, I would like to say thanks to the opponents for a very fruitful discussion, and then also guiding me in what to do next, apart from my own ideas. Apart from that, I would like to thank my supervisors who have been wonderful, and it's very rare to find supervisors that you match with frequency, especially the ones that work on the last day before deadlines. Yeah. I would also like to thank my colleagues that we have a very good microenvironment that is conducive for research in our facility, and my family who has always supported me, and my wife because she knows she is very special to me. Thank you.
Okay, thank you, Harsh. The public defense is now concluded. The university board will be informed of the committee's decision concerning the public lecture and disputation. On behalf of the university, I will then say thank you to the candidate, to the opponents, and to the audience. And the audience may now turn on their microphones either to congratulate Harsh orally, or you may congratulate him via the chat function. Thanks a lot.
Thank you.
Yeah, hello Harsh. Congrats from Hurvaye. You did a great job. Actually, I didn't do much, but I listened, and you had a pretty good defense. So congratulations.
Thank you very much.
Yeah, congratulations, Harsh. I really enjoyed talking to you and discussing your work. You really know what you're talking about. So it was a very nice discussion to have. And I hope we can meet later on when we are allowed to move to different countries and actually discuss our work. I'm looking forward to that. You did very well.
It would be interesting to maybe collaborate in future and see how it goes. So it was great having you as an opponent, Elsa.
Thanks.
Well, of course, also congratulations from me, Harsh. And well, we'll see what will happen. Yeah? Maybe you can continue your work on vulvar carcinoma, and we see one another again.
Yeah. Thank you very much, Harry. Yeah. It was good to have you as an opponent. And thank you for some of the information or advices on the pathological conditions. Thank you.
This goes vice versa. Reading your thesis was also good for me. Thank you.
You should read the chats. They are just popping up now.
Yep. I would do that. Yeah. I will reply to them on the chat maybe.
Congratulations, Harsh. It was a great experience following you today.
Thank you, Chetan. Thank you for all your help in this difficult time, actually. So thank you.
Anders, I think we have finished.
Yeah, so we are all finished. I'm just writing some congratulations here in the chat field. So you can now, those of you are, you can turn off or leave the meeting. And thanks again.
Okay. See you
Thank you very
again.
much.
Bye bye.
Bye bye.
Bye.